My name is Siyuan Wang, and people also know me as Stephen Wang. My lab at Yale Genetics develops state-of-the-art image-based spatial omics technologies to understand three-dimensional genome organization and the spatial transcriptome, and how they impact cellular functions in complex tissues in aging and diseases. For a historical look at techniques that we and others have developed to visualize the 3D genome, please check out our video, A Brief History of Contemporary 3D Genomics Technologies, available from the Genome TTCC YouTube channel. For additional content, please also check out our other publication highlight video on the perturb tracing technology, another exciting work recently coming from the lab. In today's talk, I'm going to introduce our new flagship image-based spatial transcriptomics technology that enables sequencing-free whole genome spatial transcriptomics at single molecule resolution. This work was co-led by my postdocs Yubao Cheng, Shengyuan Dang, and Yuan Zhang, and was funded by NIH, particularly the National Human Genome Research Institute, NHGRI, and the Pershing Square Sloan Cancer Research Alliance. There are currently two major classes of spatial transcriptomics technologies. First, there are spatial capture or tagging-based spatial transcriptomics. These techniques uses a 2D array of oligos carrying X and Y barcode sequences to label transcripts from a tissue section, and then use in vitro high-throughput sequencing to read out both the barcode sequence and the transcriptal sequence. The advantage here is a high genome coverage, and in principle, all poly-A transcript species have a chance to be tagged and sequenced. However, these techniques have limited spatial resolution, which is defined by the size of the 2D array element. They don't have single molecule spatial resolution, and oftentimes the single cell segmentation in space is also ambiguous, leading to challenges in fine-scale spatial analysis. Then there are these image-based spatial transcriptomics techniques, including multiplexed fluorescence in situ hybridization techniques such as RAEFISH that I co-invented before, and the in situ sequencing techniques. By imaging, these techniques have single molecule spatial resolution. Cell segmentation is precise and unambiguous, allowing fine-scale analysis, but they require researchers to pre-select a few hundreds to a couple of thousands transcript species of interest to target, and they don't have genome-wide coverage. Due to this limitation, image-based spatial transcriptomic studies sometimes only yield validations of prior knowledge rather than new insights due to the pre-selected RNA targets being well studied. So an ideal technique should have both the genome-wide coverage and the single-molecule spatial resolution. Here, we set out to develop such a technique based on imaging that by default have the single-molecule spatial resolution, and the challenge here is to increase the coverage. So why previous transcriptome imaging techniques cannot reach 20,000 genes? For RAEFISH, it's because it needs to tile each RNA with tens of probes, so it's not good for short RNAs. For in situ sequencing techniques, one of the reasons is because the padlock probe design in these techniques are not compatible with cost-efficient probe library synthesis. In situ sequencing techniques rely on these padlock probes and rolling circle amplification to amplify a short target sequence so they can actually probe short RNAs. However, these padlock probes need to be individually synthesized, which is cost prohibitive when scaling up to target the whole transcriptome. In RAEFISH, there is actually this cost-effective probe library amplification procedure, but the key constraint here is the 5' end of the final probe will have a long constant region. So this is compatible with RAEFISH, but not compatible with the padlock probe designs in situ sequencing, because in these padlock probe designs, the 5' end of the padlock probe is either completely variable or only have a very short constant region. And because of these, the probes in the in situ sequencing techniques needs to be individually synthesized, and if one wants to cover 20,000 genes, the probe cost alone would be tens of thousands of dollars per replicate. So here we develop a new technique called RAEFISH, And in this invention, we created padlock probe designs that allow the 5' long constant region, thus compatible with the cost-effective probe synthesis procedure. This reduced the probe cost by more than 100-fold and allows genome-wide coverage. So in RAEFISH, we hybridize these cheap reverse padlock and priming probes to our RNA targets in fixed and permeabilized cells, and the padlock probes are then ligated with the help of a splint oligo. Then we perform rolling circle amplification. Finally, we use the RAEFISH to encode and decode this amplified target sequence on the rolling circle amplicon. This RAEFISH encoding and decoding procedure also allows us to spread the RNA signal to many more imaging rounds than in situ sequencing, and this helped us avoid a signal crowding issue when imaging the entire transcriptome. In sum, RAEFISH combines the strengths of in situ sequencing and RAEFISH to enable genome-wide targeting in image-based spatial transcriptomics. It creates a design to allow short transcript targeting, cost-effective probe synthesis, and RAEFISH-based decoding to minimize signal overlap. Does this work in cells? Here we first designed a RAEFISH probe library targeting 23,000 human genes, including nearly the entire protein-coding transcriptome and 7,000 long non-coding RNAs. We performed RAEFISH with this library in A549 human cancer cell line, and here each individual dot is an RNA molecule, and we can decode their gene identity. On average, we detected nearly 4,000 RNA molecules per cell from about 1,300 genes per cell. This detection efficiency is comparable to single-cell RNA sequencing and high-quality in situ sequencing methods. At the bulk level, RAEFISH RNA copy numbers of different genes correlated well with their bulk RNA sequencing results, validating the RAEFISH result. Can RAEFISH be applied to tissue? Here we designed a second RAEFISH probe library targeting 22,000 mouse genes, including again nearly the entire protein-coding transcriptome and 5,000 long non-coding RNAs. We perform RAEFISH with this library in mouse liver tissue sections. Using the single-cell RNA profiles, we can easily distinguish the cell types in liver based on their liver cell type marker gene expression. Here we projected the identified cell identities back onto the tissue image. Note, this is a very zoomed out large tissue image, and here each dot represents a cell and color-coded by the cell type identity. Here across this liver tissue, we see the periportal hepatocyte showing in red and pericentral hepatocyte showing in cyan, showing an expected large-scale zonation pattern in liver, again validating the technique in tissue application. As one example of the many analysis and discoveries we performed, at a finer scale in liver, we asked which liver cell types are preferentially interacting with each other. For this, we analyzed which cell types are more likely to be neighbors than expected from their abundance. We found strikingly cholangiocytes preferentially interact with leukocytes. Cholangiocytes are the epithelial cells lining bile ducts in liver, and leukocytes are the immune cells in our blood. So this interaction indicates cholangiocytes may play an immune function. Indeed, when we further analyzed the differentially expressed genes in cholangiocyte neighboring leukocyte versus those cholangiocytes not neighboring leukocytes, we found several upregulated genes in the cholangiocyte neighboring leukocytes are components or chaperones of the MHC class II complex, which is used for professional antigen presentation. We can also do the mirroring analysis and ask, what genes are differentially expressed in leukocytes neighboring versus not neighboring cholangiocytes? Also, what subtypes of leukocytes are preferentially neighboring cholangiocytes? We found upregulated genes in leukocytes neighboring cholangiocytes are mostly cell surface receptors or extracellular matrix components, indicating these adhesion molecules expressed from a subset of leukocytes may mediate the leukocyte-cholangiocyte interaction. To distinguish which subtypes of leukocytes preferentially interact with cholangiocytes, we found particularly some neutrophil markers are preferentially upregulated in the leukocytes neighboring cholangiocytes. And these results suggest that neutrophils expressing certain extracellular matrix proteins and surface receptors preferentially interact with MHC class II expressing cholangiocytes in liver. We similarly applied RAEFISH in mouse placenta and lymph node tissue sections and generated similar spatial-dependent gene expression observation at different spatial length scales in a hypothesis-free manner. Finally, in this work, we also developed perturbed RAEFISH that enables direct readout of gRNAs in an image-based CRISPR screen. Previous imaging CRISPR screens often need to pair each guide RNA with a barcode RNA that's easier to detect, but the pairing can be shuffled by RNA recombination during the packaging of lentivirus used in such screens. This shuffling can limit screen accuracy and sensitivity. With RAEFISH, we no longer need a barcode RNA in such screen. So here we developed perturbed RAEFISH to directly read out the gRNAs, and we can see the gRNA identities in individual cells can be readily decoded. We performed a pooled CRISPR knockout screen, individually knocking out about 600 expressed genes with 3 guide RNA for each target gene and additional non-targeting or intergenic control gRNAs. For high-content phenotyping, we used RNA merfish to image the expression of about 500 different RNA species. Here, by clustering perturbation genes that generated a similar transcriptome response, we found gene clusters showing similar perturbation responses. And the genes in these clusters enrich specific gene ontology terms. There are also under-annotated genes in these clusters, and now we can hypothesize about their potential functions based on their clustering with the better studied genes. As a summary of this study, RAEFISH addresses the major compromises of previous spatial transcriptomics technologies by simultaneously enabling single molecule spatial resolution and whole genome level coverage of long and short endogenous and engineered RNA species in cell cultures and intact tissues. This work represents the first time that more than 20,000 RNA species were directly probed and imaged in situ with any technology, and the first time numerous guide RNAs were directly probed and distinguished by imaging in a high-content CRISPR screen. While not all technical aspects of this work were discussed here, please find the full paper through the link in the description. Processed imaging data and original codes are also available for download at the links in the description. The next steps of RAEFISH development include combining RAEFISH with tissue clearing to allow thick tissue profiling, further increasing the detection efficiency and coverage of RAEFISH, and developing in vivo applications of perturbed RAEFISH. To access the RAEFISH technology, a key requirement is to have an automated imaging and fluidics platform. We welcome researchers who are interested in setting this up to reach out to us, and we will provide instructions on several potential options of this setup. Thank you for listening.